Hi, Dr. Wang here. Today we're going to go over this assignment called Flowchart Lab 3. This is a lab for restriction digestion of the plasmid DNA. In the previous lab, Lab 2, we have virtually isolated 3 plasmid samples. So today we're going to determine their identity. If you go to the comments page, go to MSH2 project overview, I would highly recommend that you watch the second video right here, MSH2 project part 1. I'm going to go over some of the information in this video very quickly. For more detailed information, please go over this video. Right, so coming back to the 3 plasmid DNA that we have isolated in the previous lab, one of those is called PMSH2. This is a circular plasmid DNA containing the coding region illustrated by this purple line. That is the coding region for MSH2, the wild-type version. And this plasmid right here also contains the MSH2 coding region, but with a point mutation. So this is the mutated version. And this plasmid over here doesn't contain any MSH2 insert. And there are techniques that allow us to distinguish the 3 plasmid DNA from each other. And in this lab, we're going to use restriction digestion. The 2 enzymes that we're going to use is called XBAR1 and APA1. So if you go to the protocol for this week, the detailed information for these 2 enzymes can be found in the manual for these 2 enzymes. All right, so this is our starting material. So we're going to use these 2 enzymes to cut the plasmid DNA into pieces. Restriction enzymes recognize specific sequences on the DNA and then cut the DNA open. All right, so here, if we look at this empty vector PRS413, we can see the 2 enzymes cutting site are very close to each other. So this cut is going to generate a large fragment and a tiny piece. If we look at plasmid DNA that contain MSH2 insert, we can see this digestion is going to give us this large piece along with a smaller piece that is the MSH2 coding sequence. And because this purple, because this purple piece is larger than this tiny piece, we can run the digestion product on agarose gel, which separate DNA by size, and we can actually tell the difference between the empty vector, right? So right here, this is this tiny piece right here, and then the MSH2 insert is right here. So this technique that we're going to do in lab 3 and 4 will allow us to distinguish the empty vector from plasmid that carry the MSH2 insert. So how do we set up this restriction digestion reaction? This is the calculation listed in flowchart lab 3. So this assignment is right here. So I'm going to click on this. So the step-by-step protocol for this lab is right here, and I have rendered this protocol into a flowchart. This is a fairly complicated calculation, so I would strongly recommend that you form group to tackle this assignment. All right, so this entire setup can be divided into 3 steps. The first step is to make diluted plasmid DNA. So here we can see that this is described in the protocol. The first step is to dilute the original sample with water to make 12 microliter diluted sample. Right, so this is right here in this flowchart, step 1. So we're going to do this for all 3 samples. So what we can do is to do 1 sample at a time. So I'm going to isolate this step right here. All right, so we take the original sample A, so with the concentration that's measured by nanodrop. So this is our starting concentration, right? So this is our first concentration. I'm going to call the solution 1 as the starting material, and this diluted sample A is going to be our end result. So I'm going to call this solution 2. So the concentration of the original sample A is 604.2 nanogram per microliter, and then we are going to use a microliter of it. So this is the volume for the first solution. And then the end product solution 2, the concentration C2 is 100 nanogram per microliter, and the volume is 12 microliter, right? So the concentration and volume of this solution 2 is specified in the protocol, 12 microliter of the diluted DNA at 100 nanogram per microliter. So as you can see here, this is a typical C1, V1, C2, V2 question, right? So C1, V1 equals C2, V2. All right, so, and then we can plug in all the values, right? So 0.2 nanogram per microliter multiply a microliter should equals 100 nanogram per microliter multiply 12 microliter. When you plug in values, it's important to plug in the associated units as well. All right, so here, cancel unit, and then we can solve A. Right, so the value for A is the answer for the first question right here in this assignment, right? So what is A? Please include 2 decimal points. Okay, so once we have solved A, we're going to solve the value for B, the volume of the water to use. So here we can see when we take a microliter of the original plasmid sample and then add B microliter of water, we're going to get 12 microliter of the diluted sample. So A plus B equals 12. So when you know the value for A, you can easily solve the value for B, and that's the answer for the second question. And if you repeat the step for sample B and sample C, you're going to get the answers for questions 3, 4, 5, and 6. And this completes step 1, diluting all 3 plasmid samples to 100 nanogram per microliter. Step 2 is to prepare master mix. This is a fairly new concept. So what is a master mix? Let's look at the entire flowchart. We need to assemble 6 reactions total. So the digested reaction, so let me go back to the vertical right here. Right, so we need to assemble 3 digested reaction, 20 microliter each, that contain the following ingredients. We also need to assemble 3 control reactions called undigested that contains all the ingredients similar as above, except for the 2 enzymes. All right, so in total, 6 reactions. We can, of course, assemble those reactions one by one and do that 6 times, or we can assemble master mix. So what is master mix? Right, so I'm going to use cooking as an example. So for example, if you cook a large pot of soup for a number of guests, right? So there you can of course cook them individually, right? Or you can cook a large pot, right? So I have a large pot of soup right here that contain all the common ingredients that everyone has agreed to eat, right? So water, of course, maybe some meat, some rice, some beans. Once you finish cooking this large pot, you can ladle them into individual bowls, right? So in molecular biology, we call this a liquid. We can aliquot the soup to individual portions, and then this is when you can add unique ingredients. You can add chili powders if this guest like spicy food, or you can add onions, or you can add cilantro, and etc. Right? So those are unique ingredients that that different guests have different preferences. Right? So you don't want to add unique ingredients into this large pot. You want to add them later. So this large pot right here is called master mix. So here we have 2 tubes on this flowchart representing the master mix for the all digested reactions and another one for all the undigested reactions. We can see that what goes into the master mix are the common ingredients, the enzyme, the buffer for the enzyme, and water. And similarly here for the undigested, what goes into here are buffer and water. All right, so once you make the master mix that contain all the common ingredients, you're going to add a certain portion of it to an individual reaction, and then you're going to add a unique ingredient. As you can see here, sample A only goes to sample A, either digested or undigested. Right, so the plasmid DNA here are the unique ingredients because it only goes to here and here and not here, here, or here, here for sample A. Right? So how do we finish the calculation for master mix? So I have all the information listed down here. So let me come back to the protocol right here. Right, so to assemble 20 microliter, blah, 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 right? So I have all the information listed here, right? So the total volume for a single reaction is supposed to be 20 microliter. And the concentration for those ingredients are 1x buffer, 5% for both enzymes, and then 500 nanogram DNA. And our starting materials are listed right here. Okay, so this is the starting material. We have 10x buffer and we have pure enzyme. So here our starting material 10x buffer and 100% pure XBA1 and APA1. Our starting material for the plasmid DNA is the diluted sample we have already prepped. So this is 100 nanogram per microliter. All right, so how do we figure out how many microliter of the buffer to use? Right, so for example, so this calculation can be done this way. All right, so let me clear some space here so we can do this calculation. All right, so for the ingredient called buffer, we can set up the equation this way. So our starting material is 10x, right? So this is our C1 starting concentration. And we're going to use question mark microliter of it. So this is our volume for the starting material. And then it's going to equal the concentration in the final reaction. So this is 1x. And then volume of a single reaction, this is 20 microliter, right? So 20 microliter right here. So once we have set up the equation, we can cancel unit. So X, this is an artificial unit. The working concentration is 1x and the stock concentration is 10x or 10 times concentrated. Microliter can be canceled. And now we can solve question mark, right? So once you solve question mark, this is the amount of 10x buffer to use in a single reaction. Right. So, and then for the same thing, we can repeat the process for the second ingredient called XBA1, right? So our initial concentration is 100%. Our volume, initial volume is X microliter. Our second concentration, C2, this is 5%, and the V2 is 20 microliter. So a very similar process, and you can solve the volume for XBA1, similarly for APA1. All right, so now we are going to solve plasmid DNA. How much plasmid DNA should we use? So plasmid DNA is a unique ingredient. So in a single reaction, we're supposed to use 500 nanogram. Right? So gram is a measurement of mass. So this is mass. And then our starting material is 100 nanogram per microliter. So this is our initial concentration. And then we're going to figure out how much of it to use. So this is our V1. And then here the equation is C multiply V should equal to the mass of the solute. So here, similarly, we can cancel unit first and then solve X. All right, so this value will go to here. So once we have solved the volume for 4 of the 5 ingredients, the volume for water is literally, once you add up everything, it should be 20, right? So once you have the value for this 4, you should be able to solve the value for water. So now we have figured out how to assemble a single reaction, but we're not going to assemble a single reaction. We're going to assemble a master mix. So this is step 2. All right, so step 2, we are going to prepare enough master mix for 3.5 reactions. We can see that this master mix will be used to assemble 3 reactions. Instead of assemble 3 reactions, we just make a little bit more, right? Just like cooking, if you have 3 guests, you probably don't want to cook just for 3. You want to cook a little bit more just in case. Right? So we're going to assemble enough for 3.5 reactions instead of 3. So what you can do here, so what this value is you take this value, right, so the question mark that you have already solved, and then you're going to multiply this value by 3.5, and then this is going to give you the amount of 10x buffer to use in the master mix. So if we look at the flow chart, this is value I, right? So the amount of 10x buffer to use in this master mix digested. So here, this is value I. Okay, so and similarly, you can take X bar 1, right, so do the same, and take APA 1, do the same, take water, do the same, and then you're going to get value, so let me come back, right, so X bar 1 is value G, APA 1, this is value H, and then water is value J. Right, so 4 common ingredients goes to the master mix. Right, so this is the master mix digested, and then those values are the answer for question 7 all the way to 10. Okay, so here, pay attention to the step. Plasmid DNA is a unique ingredient, so this should not be included in the master mix, right? So if you add chili powder to the entire pot, then basically you have this ingredient that not everyone like in every bowl now, right? So you cannot add unique ingredient into the master mix, right? So this one doesn't apply. I'm going to cross this over. So now coming back to how do you assemble, oops, sorry, how do you assemble the master mix undigested? So only 2 ingredients goes to here. The 2 enzymes do not go into this undigested master mix. So here it only contains buffer and water. So basically the same step, right? So whatever you did over here, you're going to do the same thing over here. The only difference right here is the amount of water would be different. Sorry, let me cross this too. So it only contains buffer and water. So everything adds up to, oh, sorry, and DNA. So everything adds up to be 20. So the amount of water that you use here is going to be different from this value here. So this is what I want to point out. All right, so you can finish the entire calculation and then figure out the value for K and L. So now we have completed step 2, right? So we have the 2 master mixes prepared, and we have the 3 diluted plasmid DNA prepared. So now it's when we're going to assemble the individual reactions. So we can see the individual reaction, for example, let's look at this guy over here, is by mixing M microliter of the master mix with O microliter of the diluted sample. All right, so how do we solve the value for O? And you actually already solved it. It's right here, right? So the value of O is right here. This is the volume of this unique ingredient used in a single reaction, right? So this is the value for O, and it's actually going to be the same for P and Q. Those 3 diluted samples are at the same concentration, right? So you're going to use the same volume for all 3 samples. And this is actually why we diluted all 3 samples to the same concentration in the first step. Right, so OPQ, right, so this is right here, right? So this is the value for the last question, right? So what about the volume of the master mix in a single reaction? So for this, we're going to come back to right here, right? So you already solved the volume of all the ingredients that goes into master mix, right? So you're going to take these 4 values, add them up, We're going to take a sum of the 4 values. So this is the volume of the master mix. And then you're going to you're going to divide this by 3.5 because this is enough for 3.5 reactions. So if you divide this value by 3.5, this is going to give you the volume of the master mix in a single reaction. And then this is the value for M. And you can do the same for N, and you're going to figure out that M and N are actually equal to each other. So this is the answer for question 13. All right, so here we have completed the flowchart preparation for this lab.